Welcome friends, let us continue our lecture in this course Microbial Biotechnology. We are in module 7 and in this particular lecture we will be speaking about microbial biocontrol. So, which is divided into various sections. We will be speaking about synthetic pesticides and fertilizers and how they are not that suitable, and then the importance of biocontrol, suitable sustainable pest management and disadvantages of biocontrol, and also the different biocontrol methods adopted. And we have another two sections, 3 sections. We will be discussing about these sections details as we come to them in the course of time. So, before we start, let us revisit overall in the plant microbiome composition, which is quite complex and diverse. And as you can see in this picture, we have these microbes which are ubiquitous, present almost everywhere in the leaf, in the soil, and maybe in the fruits and flowers. And you can see the various counts of them in endosphere. You have as many as 10 to the power -4 CFU per gram, and then you have in phylosphere similar number, 10 to the power 5 to even 10 to the power 6. And then you have them in rhizosphere, and then we have other organisms like nematodes, and then also arthropods and virus particles. So, a plant is exposed to so many different kind of organisms, microorganisms, and little bit of submicroscopic particles like virus, and also higher organisms like arthropods. Many of these are beneficial and some of them are harmful to the plant ecosystem. To control the harmful organisms, we use synthetic pesticides, and also we use synthetic fertilizers for helping boosting up plant nutrition. But then in the long term use of these pesticides and artificial fertilizers, it has been found that they are harmful and are not sustainable. For example, they are contributing to health risks for humans, such as they cause respiratory problems, skin irritation, long term effects like cancer. And the agricultural workers who apply them and work on those fields which are contaminated with these pesticides and artificial fertilizers are having chronic exposure and thereby are the most vulnerable group. And even as a consumer, the pesticide residue in the food product, agricultural plant food product, it is causing health risk to the consumer as well. And the continuous application of pesticides will result in resistance development and thereby reduce effectiveness over time, and then farmers try to overcome this resistance by increasing the dose of the pesticides, which actually is much more harmful in the long term. So, these artificial fertilizers and pesticides impact the environment in various ways by polluting the soil and the water, and finally that harms the plants, animals, people, and the soil microorganisms. And one of the problem with these pesticides are that they harm non-target organisms. We are trying to target a particular disease causing organisms or a particular insect which is a pest, but then when we apply these, they may be broad spectrum and they will kill many beneficial insects as well as microbes, and also they may be toxic to animals and then therefore harm non-target organisms. So, in order to overcome many of these shortcomings, one of the idea is to use biocontrol as a pest management strategy, which will employ natural enemies or organisms to manage and reduce harmful pests, disease and weeds in agriculture and natural ecosystems. So, here you can see these Polistis webs, which is trying to feed on some of the harmful organisms colonizing cotton. So, biocontrol minimizes reliance on chemical pesticides and fosters a more eco-friendly and sustainable approach to pest management. So, what are the importance in brief of biocontrol for sustainable pest management? Number one is there is less chemical use. By using natural predators and other organisms, farmers can use fewer synthetic pesticides. And this is important because too much pesticide can harm the environment and other animals that are not pests, and also us, the humans. Then it provides long-term solutions. Biological control can help keep pest populations low over a long time. And instead of just killing pests quickly, it helps create a balance in the ecosystem, which means that pests are kept under control naturally. And it promotes biodiversity by introducing or enhancing the populations of beneficial organisms. Biological control helps increase the variety of life in a particular area, and this is beneficial for the environment because a diverse ecosystem is usually healthier and much more resilient. Biocontrol agents are essential for sustainable and effective pest disease management. There is no doubt in it, and it offers significant benefits in both agriculture and environmental context, and it has various merits or advantages. For example, it helps in the pest and disease management, and then it has targeted actions: do not kill non-target organisms, enhance agricultural sustainability, safety and health benefits, do not harm human or animal, and it helps in economic benefit and reduced environmental impact and resilience to environmental changes. So, we have direct and indirect mechanisms of biocontrol which involve interaction between bacterial biocontrol agent, pathogen and plant, for example. So, here when we apply a bacterial biocontrol agent, it will help directly by induction of resistance, and then also by plant growth promotion indirectly. Then we have direct action where it will kill the pathogen. So, antiviral microbial metabolites and competitive exclusion and quorum sensing interference will help us directly in controlling the pathogen, either by killing it or restricting it. Although biocontrol offers many advantages, it also suffers from potential challenges and limitations. We must move beyond denial and recognize these challenges to develop more effective biocontrol strategies and ensure that their benefit outweigh any potential drawbacks. Some of the disadvantages are variability in effectiveness. So, there may be batch to batch variation, or there may be also variation due to environmental conditions. Then it is very complex when it comes to application or implementation, and there can be potential non-target effects, although we have told that it overcomes the disadvantage of synthetic pesticides. To some extent there may be potential non-target effects which need to be examined and optimized. Cost and availability may sometimes be high because we need to grow these biocontrol agents under artificial conditions, and there is chances of resistance development as well in this case if we are not following standard operating procedures or package of practices. There can be also certain regulatory and legal issues which do not allow the deployment of certain biocontrol agents in certain countries or certain areas and localities, and overall there is a challenge of integration, particularly when it comes to integrated pest management. Nevertheless, it has been found to be very, very effective because the disadvantages are actually, compared to the advantages, are much lesser. So, there are various methods of biological control. If you look into the primary methods, number one is conservation of existing natural enemies. So, every ecological niche or agro-ecosystem may have naturally present enemies which we need to preserve and enhance and enrich. Another method is introducing new natural enemies into a agro ecosystem or an environmental niche, which is a most practiced method. So we call it as a classical biological control. And then there is mass rearing and periodic release. So we will rear them in artificial conditions and keep on releasing these enemies of the various pests. So how do we conserve existing natural enemies? For example, we have here two examples, the natural parasitoids, Trichogramma and Brachymeria, and then we have here natural predators, the Coccinellid and lacewing. Conserving natural enemies is a key strategy in protecting our ecosystems and ensuring a sustainable future for our planet. Natural enemies such as predators, parasitoids and pathogens are crucial for controlling pest population and reducing the need for chemicals. By protecting and encouraging these beneficial organisms, we improve pest control, support biodiversity, strengthen ecosystems and promote sustainable farming. Natural enemies also provide important services like pollination and nutrient cycling, making our agricultural system more eco-friendly and resilient to climate change. These overall reduce the production cost and pesticide pollution and helps prevent pesticide resistance. How do we provide a habitat for natural enemies, which is a vital strategy in integrated pest management? We need to create diverse environments, for example, like hedgerows and cover crops attract beneficial organisms that naturally control pests, reducing the need for chemical pesticides and promoting biodiversity, pollinator support and sustainable farming. Natural enemies require resources like pollen, nectar and prey to thrive. Habitat design should consider both pest and natural enemy behaviors. For example, native lady beetles prey on Colorado potato beetle eggs, but also rely on aphids and pollen, though further research is needed to optimize their use in pest control. Now let us look into the method of classical biological control, where we rely on introducing new natural enemies. So this is done intentionally by releasing non-native species that can naturally control invasive species or pests. We begin with identifying the target pest, understand the pest's biology and the ecological role. Then we select the natural enemies for its effectiveness as a predator, then parasitoid or pathogens from the pest's native range. Then we need to follow quarantine and also undergo testing, assess the potential natural enemies to ensure they won't harm non-target species. And then rearing, cultivate a sufficient population of the selected natural enemies, release and introduce the natural enemies into the pest infested area at the right time. Monitoring, continuously track the population dynamics of both the natural enemy and the target pest. While doing this, we need to observe certain precautions, like we need to carry out ecological impact assessment, evaluate the potential effects of non-target species and ecosystems. And then we need to know the host specificity, ensure the natural enemy is highly specific to the target host and do not target non-target organisms. We need to follow regulatory compliance at the local, national, international level regarding non-native species introductions. Then we need to have continuous public engagement, whereby we inform and involve stakeholders to gain support and address corners and make them partners in the entire exercise. Then we always should have contingent planning for preparing for possible negative outcomes such as invasiveness. And then long-term monitoring is very, very essential. We implement ongoing assessment to evaluate effectiveness and ecological impact. Some of the successful examples of controlling the alfalfa weevil with introduced parasitoids can be seen in this picture. You have the adults of Oomyzus and attacking an alfalfa weevil larva, and then you have the pupa of these Oomyzus dissected from a mummy formed from the integument of the alfalfa weevil larva. Then in this case you have the Bathyplectus anurus. These are adults, are parasitoids of alfalfa weevil larva. Mass rearing and periodic release. So here seasonal inoculative release is one of the method. This is useful when natural enemies cannot overwinter, requiring annual reintroduction. So we have introduced them, but then due to change in weather, lowering temperature, they will all die. So we need to introduce them again next season. So there are case studies in European greenhouses for controlling pest resistance to insecticides, and here we have the example of Pediobius against Mexican bean beetles, and Idovum putuleri against Colorado potato beetle. Then we also do biological insecticides and inundative release. This focus on immediate pest reduction rather than establishing long-term natural enemy populations. So we have biological pesticides like Bacillus thuringiensis and beneficial nematodes. They target specific pests without creating lasting populations of natural enemies. In inundative release, we go for mass releasing natural enemies such as lady beetles or parasitic webs for rapid control, but these do not establish sustained populations. While inundative release can be costly, ongoing research aims to optimize rearing methods and reduce expenses, enhancing its accessibility in integrated pest management. Let us now discuss about biocontrol agents. So biocontrol agents, as you have understood by now, are living organisms such as microorganisms like bacteria, fungi, yeast, or even viruses, which are not living organisms of course, and they are used to manage harmful pests and pathogens in agriculture. They work by reducing the pest or pathogens population or impact through mechanisms such as antibiosis, competition, parasitism, or inducing host resistance. We will be discussing each and every one of them in the next few slides. Biocontrol agents are a key part of IPM, providing an eco-friendly alternative to chemical pesticides and reducing the risk of resistance. So some examples are like Trichoderma fungi and then Bacillus bacteria, and then we also have Bacteriophages, which are viruses. What are the various types of biocontrol agents? They may be predators like lady beetles, which actively consume pests. Then we have parasitoids like parasitic webs, which kill hosts by laying eggs inside them. Then we have pathogens like Bacillus thuringiensis, which infect and kill pests, and competitors like Pseudomonas fluorescens, which compete with pathogens, and cover crops compete with weeds for resources. These agents are key components of integrated pest management, offering targeted and sustainable pest control. Our discussion will be focused more on the microbial agents because this course is on microbial biotechnology. So we have predators, and then we have parasitoids, we have pathogens, and we have competitors, as we have discussed in the earlier cases. So when it comes to predators, these are animals that hunt and eat other animals. In this context, we are talking about small creatures like lady beetles and lacewings. These predators are called free living because they do not rely on a host or a specific environment to survive. They can move around freely. Insect predators can be found in many places on a plant. They can be on the leaves, stems, and even in the soil below the plant. This means they can hunt for food in various environments, making them very adaptable. So we have certain specialized predators which are very choosy or picky about what they eat. They will only hunt specific types of pests. For instance, a specialized predator might eat only aphids and ignore the other insects. Then there are generalist predators which are not choosy. They will eat a wide variety of insects, including both pests and other beneficial insects. This means they can help control many different pest populations and also may be harmful when it comes to consumption of beneficial insects. So we have some of these examples like the rope beetle, Then we have the hoverfly and syrphid fly, and then we have the minute pirate bug or insidious flower bug. The next type is the parasitoids. These are insects that lay eggs on or inside another insect. During the immature stage, parasitoids feed on the host's body fluids and organs. Once the immature parasitoid is ready to become an adult, it leaves the host, often killing it in the process. They are very specific about which insect they can use as host, meaning they can only develop on certain types of insects. Because of this, it is very important to correctly identify both the host and insect parasitoid to use them effectively in controlling pests. So there may be two main types. One is the endoparasitoids, which begin their life inside the host, where an adult female lays eggs or larvae, and then the young feed on the host from within, ultimately killing it as they grow and eventually leave the host body. And then there is the ectoparasites, live on the outside of the host, attaching themselves to the host body. They feed on sucking out the body fluids through the host skin. While they harm the host, they typically do not kill it as quickly as endoparasitoids do. So some examples are the Aphidius or Colomini, a small parasitic webs that attacks many types of aphids. Is mostly active in warm temperatures and can lay up to 200 eggs after many attempts during its life. Then you have the endoparasitoids. For example, the greenhouse whitefly is a harmful pest for plants like cucumbers and tomatoes, laying eggs on the underside of leaves, while whiteflies harm plants by sucking sap and can spread diseases. So controlling their population becomes very important. Whitefly parasite, Encarsia formosa, lays many eggs in them, killing the larvae before they grow up, and the adults also feed on the juices from the larvae. The next class are the pathogens. These are tiny organisms like bacteria, fungi, and virus that can make insects sick. These diseases can slow down how fast insects eat and grow, stop them from reproducing, or even kill them. Some nematodes, which are tiny worm-like creatures, can also infect insects and cause disease. They work together with bacteria to harm the insects. So we have here the example of Trichoderma species, which effectively control root rot in sunflower caused by Fusarium solani. When there are many insects in one area, diseases can spread quickly among them, especially if the environment is right for the pathogen to thrive. Antagonistic microorganisms are also used to combat plant diseases, especially those caused by soil-borne pathogens, which are challenging to control. These microorganisms work through mechanisms such as competition, antibiosis, suppression, and hyperparasitism. Fungal biocontrol. Fungi are used to control harmful pests and pathogens in agriculture and horticulture. Fungi either directly attack pests or pathogens or outcompete them, offering an eco-friendly alternative to chemical pesticides. Some of the examples of fungal biocontrol agents are Trichoderma species. These are widely used for controlling soil-borne plant pathogens such as Fusarium, Rhizoctonia, and Phytophthora. They work through competition, antibiosis, and parasitism. Beauveria bassiana, it is an entomopathogenic fungus that infects and kills a variety of insect pests like aphids, beetles, and caterpillars. Then we have the Metarhizium anisophilia. These targets various insect pests including termites and locusts by infecting and killing them. Then there is Gliocladium virens. This is effective against damping of diseases caused by fungi in seedlings. Then we have others like Oreobasidium pullulans. These fungus combats pathogens like Botrytis cinerea and Penicillium species through nutrient competition and antifungal metabolite production, effectively protecting fruits like apples and grapes and extending their shelf life. We have also Candida oleophila. These yeasts prevents rot in citrus and apples by competing for nutrients and inducing host resistance against pathogens like Penicillium species. It offers a non-toxic solution for post-harvest protection. Others such as Saccharomyces cerevisiae and Pichia anomala also produce antimicrobial compounds and enhance plant defenses, making them promising alternatives to chemical pesticides for controlling fungal diseases and post-harvest spoilage. So, what are the advantages of using fungi as biocontrol agent? Number one, they are eco-friendly. Fungal agents are natural and degrade harmlessly in environment, reducing chemical pollution. They have specific targeting. They typically target specific pest or pathogens, minimizing harmful to beneficial organisms like pollinators. They have reduced resistance. Pests and pathogens are less likely to develop resistance to fungal biocontrol agents compared to chemical pesticides. They help soil and plant health. Some fungi like Trichoderma also promotes plant growth and improves soil health by enhancing nutrient uptake. They are important in IPM. They are valuable components of integrated pest management strategies, contributing to long-term sustainable pest control. We have also certain virus and bacteriophages which are used in biocontrol. They serve as effective biocontrol agents, leveraging their unique properties to protect plants from harmful pathogens. They offer innovative methods for sustainable agriculture by reducing reliance on chemical pesticides. Continued research and development are essential to fully realize their potential and address application challenges. Let us discuss some of the viruses like papaya ring spot virus. This is a well documented instance of cross protection which involves using a mild strain of papaya ring spot virus. So, to safeguard papaya plants against severe strains. So, it causes some disease as you can see over here, and if we use a milder strain that will cause very mild disease or no disease, and that will actually protect the plant from further infection or colonization by a much more harmful strain. So, here we have a nitrous acid induced mild strain called as PRSV-HA51. This is derived from the Hawaiian strain, and this has been used for decades to control papaya ring spot virus through cross protection. Then we have certain, what are the functions of these particular strain? By infecting the plants with this mild strain prior to exposure to more virulent strain, the plants activate their defense mechanisms, thereby providing protection against aggressive forms of PRSV that can cause significant crop losses. This strategy has proven effective in sustaining papaya production in regions plagued by the virus. Then there is the hypovirulence virus. Here these particular virus infects the fungal pathogen Sclerotinia sclerotirum, which is responsible for white mold disease in various crops. So, when we introduce this particular virus, it reduces the virulence of this Sclerotinia species, making it less harmful to plants. This approach exemplifies a novel biocontrol method where a virus specifically targets a pathogen rather than the plant itself, thereby diminishing the pathogen's capacity to cause disease. Then we have bacteriophages, which are viruses that infect and kill specific bacterial pathogens such as Bactobacterium and Xanthomonas species. They can also prevent food spoilage caused by bacterial infections. So, some of the advantages are phages are non-toxic and highly specific to the bacterial host, minimizing harm to beneficial bacteria in the environment. However, they suffer from certain challenges like their sensitivity to environmental conditions and the need to avoid unintended effects on non-target bacterial populations. Current research aims to enhance phage durability and delivery systems. Commercial products available like agri-phage, as you can see over here, given as an example, they have been developed for agricultural use through more stable formulations, of course, are necessary for widespread applications. Now, let us discuss about the various mechanisms of actions of biocontrol agents. Some of these actually have been mentioned while discussing certain examples. Now, let us look into them in little bit of more detail. So, understanding the mechanism of biocontrol agents is crucial for developing effective formulations and obtaining regulatory approval. Despite challenges in studying complex interactions. 4 main mechanisms has been identified, which we have already heard about to some extent, like antibiosis, competition for nutrients and space. Then there is parasitism and induced resistance in host tissue. So, in this figure you can see the different methods of actions of biocontrol agents to inhibit the plant pathogens. Mechanisms involve physical contact are on the left side of the figure, and those in which physical contact is not a prerequisite are on the right side. Let us start with antibiosis. So, bacteria that produce antibiotics that inhibit or kill bacteria, and fungi serve as highly effective biocontrol agent. So, you can see here the number of strains in each species, for example, given in this diagram A. So, you can see Bacillus subtilis is the most dominating one, followed by Bacillus aminoliquefaciens, and then we have here others like Bacillus pumilus and Bacillus thuringiensis. So, different organisms have different mechanism of actions targeted by each strain. For example, some of them will be acting on the cell wall. So, for example, you have Bacillus subtilis here roughly around 22. Then some of these will be affecting the cell membrane, and some are involved in the intracellular processes, and while others offer quorum sensing. So, some of these we will be trying to discuss in some of the other slides later. So, let us start with targeting the cell wall. So, here you have this bacitracin, which prevents dephosphorylation of undecarpanyl pyrophosphate, disrupting the formations of lipid 1 and 2 and ultimately hindering the peptidoglycan layer formation. Then there is bacillicin and chlorotitani. This inhibits glucosamine 6 phosphate synthesis, which is crucial for peptidoglycan synthesis. So, those that target the plasma membrane, for example, epsilon polyl lysine disrupts membrane permeability by attaching electrostatically to the phospholipid layer. Then there is octapeptin B, which disrupts ion permeability and damages the membrane, leading to cell lysis. Then there are orantinins that cause plasma membrane lysis and the efflux of cytoplasmic metabolites. Then when it comes to targeting intracellular processes, gramicidin A and S induces the formation of reactive oxygen species inside the cell, leading to DNA and mitochondrial damage and necrosis. And bacillomycin D disrupts cell membrane and increases suppression of genes associated with ROS production and oxidative stress responses. And in targeting quorum sensing, fungicins inhibit quorum sensing, preventing communications between cells, which is necessary for biofilm formation and survival. Next comes the competition for nutrient and space. Microorganisms compete for nutrient and space with pathogens, limiting their growth. In the case of yeast, Picia goulier-mondi competes against pathogens like Penicillium digitatum, particularly in nutrient scarce environments. Then you have the Actorobacter cloacibacteria, which competes with Rhizophus stolonifer on peaches. And then yeast often form extracellular polysaccharide capsules to enhance their adhesion to surfaces like fruit skins, boosting their competitive ability. Then in the rhizosphere, there are microorganisms like Pseudomonas Jessini RU47, which compete with pathogens like Rhizoctonia solani in crops like lettuce for nutrients and space in the rhizosphere, limiting the resource essential for the pathogen's growth. And in the case of Bacillus subtilis, this particular strain SRB28 colonizes sorghum roots, forming microcolonies and enhancing root development in seedlings. The next most important mechanism is parasitism, which is a competitive interaction in which one organism derives nutrients from another The interaction is classified as hyperparasitism when the host organism is a parasite such as a plant pathogen. These phenomena is frequently observed between fungi, but reports of bacterial hyperparasitism are also there. An example is the bacterium Delovibrio bacteriovirus, which uses the cytoplasm of other gram-negative bacteria as a nutrient source. So, what are the types of hyperparasitism in fungi? We may have biotrophic mycoparasitism, or we may have necrotrophic or microparasitism. In the biotrophic, the hyperparasite feeds on a living host fungus, drawing nutrients through specialized structures called hostoria without killing the host. The interaction between host and hyperparasite is stable and balanced, although vital in natural disease suppression. This type of hyperparasitism is challenging to exploit commercially because a mass production depends on the availability of living host mycelium. In a necrotropic hyperparasitism, the hyperparasite kill host cells and then feed on them, as well as other organic matter, making them more suitable for commercial biocontrol. These fungi produce cell wall degrading enzymes such as chitinases, glucanases and proteases, which weaken the host cell wall, allowing the hyperparasite to invade. What are the prominent mycoparasites in biocontrol? Trichoderma and clonostasis are the most studied mycoparasites for biocontrol. Primarily targeting plant pathogens, they produce infection structures and kill host fungi using cell wall degrading enzymes and secondary metabolites. Enzyme production is triggered by host recognition through surface compounds like lectins and regulated by signaling pathways including MAPK cascades and cyclic AMP. Key gene families involved in mycoparasitism such as ECH42 and PRB1 in trichoderma are upregulated during host interactions, leading to further degradation of the host cell wall and its eventual death. There are synergistic gene transcription which has been observed between Trichoderma atroviridae and Fusarium oxysporum. In this figure, you can see Trichoderma atroviridae releasing volatile organic compounds, promoting plant growth and suppressing Fusarium wilt disease in tomato seedlings. Induction of resistance, promotion of the plant growth. In these, plant growth promoting microbes act as biocontrol agents by reducing plant pathogens and enhancing growth. They contribute to nutrient cycling and increase plant biomass through various mechanisms. Specific bacterial strains can suppress fungal pathogens, improving plant health, notably some plant growth promoting rhizobacteria such as Trichoderma harzianum. So, antifungal and growth promoting activities even at low concentrations. Some of the examples include Bacillus, Pseudomonas, Actinobacteria and several others. So, these PGPMs enhance nutrient uptake by various ways. Number one, nitrogen fixation. Rhizobial bacteria convert atmospheric nitrogen into a usable form, improving soil nitrogen and essential nutrients like iron and zinc. Then phosphate solubilization. Some of these convert insoluble phosphate into soluble forms for plant use. Then there is a pseudo for production. These compounds sequester iron, making it more available. Then there is hydrogen cyanide production. Certain PGPMs produce hydrogen cyanide, which inhibits pathogen growth. The mechanism of induction of resistance. Microbial biocontrol agents enhance plant defenses through two main mechanisms, induce resistance and priming. Microbial biocontrol agents trigger both induce resistance and priming by releasing signaling molecules like MAMPs, siderophores, antibiotics and volatile organic compounds. By promoting plant health without directly attacking pathogens, MBCAs help reduce the risk of resistance development in pathogens. In induced resistance, these are activated by specific stimuli recognized by plant receptors such as pathogen associated molecular patterns. Systemic acquired resistance triggered by necrotizing pathogens. SARS spreads defense signals throughout the plant and to neighbors. Then there is the systemic resistance initiated by beneficial microbes, the ISI enhances defense against various pathogens. In the case of priming, here it sensitizes plant for a quicker, stronger defense response to future attacks, offering longer lasting protection with lower energy cost than IR. There is a transgenerational priming, allows defense capabilities to be passed to offspring, which is quite an interesting phenomena. Then there is the mechanism of microorganisms producing microbe associated molecular patterns, MAMPs, recognized by plant receptors that include bacterial proteins, flagellin and recognition elongation factor TU. Then there is a fungal compounds, glucans and chitins. So in the case of the mechanism involving this defense, this induces defenses which involve producing reactive oxygen species, then phytoalexins, phenolic compounds, phytogenesis related proteins and physical barriers like cell wall and modifications. So we have various biocontrol agents which can help us in controlling pest attack on plants. So in this table we show, we list here some of the selected bacterial biocontrol agents of certain plant diseases. So you have many microorganisms listed here in this column. Then you have the target pathogens or the diseases. For example, the synonyms has been given the full form here. AC stands for Alternaria citrae. Then you have this Botrytis B, and then so on. So for understanding the full meaning of these pathogens, please refer to these footnote below. Then these particular organisms on column 1 against used in certain plant species in vivo or in trials, you can see over here, for example, orange fruits, and then you have the cherry fruits and so on. And then for reducing the disease, the dose that is to be applied is in this particular column, which is CFU per ml. So this is 20 to 70 CFU in this particular case. And then the mechanism involved is mentioned over here. Mostly these are antibiotics. Then in certain cases there is competitive exclusion, and then there is a nutrient composition, and in some certain cases it is induced resistance. So for knowing more about them, kindly please refer to the slides which will be provided to you along with this lecture material. Now, one of the important pests are the nematodes, and when it comes to biocontrol, microbes like Bacillus, Pseudomonas, Burkholderia effectively manage nematode populations by altering their behavior, including feeding and reproduction. This leads to a reduction in nematode numbers in both soil and infected roots. So Bacillus bacteria have shown significant efficacy against root knot nematodes by producing nematode suppressive compounds, and the mechanism include inducing systemic resistance through several processes like boosting enzyme activity and enhancing root exudates. So in the case of boosting enzyme activity, they increase the activity of defense related enzymes such as polyphenol oxidase, peroxidase and phenylalanine ammonia lyase. And in the case of enhancing root exudates, Bacillus modifies root exudates to include beneficial amino acids and polysaccharides, promoting overall plant health and resistance. So let us now go to section 4, where we will be discussing about traditional approaches in biocontrol, and then certain emerging paradigms in biocontrol from pure culture to microbiome engineering, and finally the development of bacterial biocontrol agents. So the traditional approach in biocontrol is to use pure culture microorganisms. So here we use single well characterized microbial strains that are isolated and cultivated in controlled environments. These strains are specifically selected for the ability to inhibit pathogens or promote plant growth. Examples are Bacillus thuringiensis and then Trichoderma species. So, in traditional approach, the various advantages are control outcome, easier to predict and manage due to well understood mechanism of action. Then specificity, we already know the targets, and it is a control of specific pest or diseases can be made a goal. But there are certain limitations because of the narrow scope. Pure cultures often have a limited range of activities and may not address complex pest and disease interactions. And there are certain adaptation issues. Single strains may not perform well under diverse environmental conditions or in the presence of varying soil and plant microbiomes. So, there is a change or shift in the paradigm in biocontrol, where instead of using pure cultures, we focus on microbiome engineering. This approach involves manipulating the entire microbial community associated with plants, including bacteria, fungi and other microbes. It focuses on enhancing or modifying the plant microbiomes to improve plant health and resilience against pests and diseases. So, we have here the comparison between individual microorganisms based reductionist approach, we are only using only one, and then the microbial community based holistic approach, which uses many. And then we are focusing currently on the microbiome engineering. So, this technique includes inoculating soils with diverse microbial communities, enhancing beneficial interactions or engineering plant microbiomes to increase their resistance to pests and diseases. So, you have here maybe first case the single isolate, then we may use biosticking, and finally the deployment of a consortia. So, what are the key strategies? Inoculation of soil and rhizosphere. Introducing a diverse range of beneficial microbes into the soil to establish a competitive and protective microbiome. The benefit of this is that it can create a more robust microbiome capable of suppressing a broader spectrum of pathogens. Then endophyte and seed microbiome engineering, where modifying the microbiome within plant tissues or seeds to enhance resistance to specific pathogens. The benefit here is that endophytes can offer systemic protection, improving plant resistance from within. Then there is the host mediated selection. Here we select microbial communities based on their interactions with specific plant hosts to enhance plant defense mechanisms. Here the benefit is that tailored microbiomes that are well adapted to specific plant types and their associated stressors. And then finally, we have the tissue and wound inoculation, where we directly apply beneficial microbes to plant tissues or wounds to provide immediate protection, and this helps us in rapid intervention in disease outbreaks or pest infestations. So, what are the advantages of microbiome engineering? It is a holistic management, addresses the entire microbial ecosystem rather than focusing on individual organisms. There is enhanced resilience, creates a more resilient plant microbe system capable of managing complex pest and disease interactions. The adaptability to varying environmental conditions is potentially more, as well as against more plant types, but there are certain challenges in the approach of microbiome engineering. The complexity, managing and understanding interactions within a diverse microbiome is more complex than working with a single microbial strain. Then stability and persistence, ensuring that engineered microbiomes remain stable and functional over time and under different environmental conditions. Then the problems of regulatory and safety issues, addressing potential mycological impacts and ensuring that engineered microbiomes do not adversely affect non-target organisms or ecosystems. So, some of the emerging directions in the shift towards microbiome engineering represents a forward-thinking approach to biocontrol, leveraging the natural interactions within microbial communities to achieve more sustainable and effective pest and disease management. So, we need to focus on optimizing microbiome design, develop methods to precisely engineer and monitor plant microbiomes for specific agricultural needs, then integrate microbiome engineering with other practices, combining with traditional pest management strategies and sustainable agricultural practices. Understanding long-term impacts and studying the long-term effects of microbial modification on plant health, productivity and ecosystem dynamics. So, here is a flow chart diagram for development of bacterial biocontrol agents. So, we have selection of antagonistic bacteria, we go for isolation and screening, then we finally select these strains which needs to be characterized, that includes identification, phenotypic, genotypic traits, mechanism of action, biocontrol efficacy in pilot test and their improvement. Once this is completed, we can go for delivery for commercial use. At this stage, we go for mass production and formulation. We need to go for biosafety and environmental impact assessment, and here specific monitoring methods are required, and of course, we need to register these biocontrol agents under regulatory approval. So, in the first case, we go for the isolation and screening of the strain selection, effective sampling from diverse niches and proper selection of samples, media and isolation techniques are crucial. At this stage, bacterial antagonists are found in plant environments and bare soil. Samples may come from suppressive soils or healthy plants in disease prone areas. Some beneficial microorganisms are rare. Isolating a large number of candidates is recommended. Techniques such as selective media and molecular markers improve the screening efficiency. In vitro test for pathogen reduction, while whole plant and ex vivo bioassays assess other mechanisms of action. A multi-pathogen approach helps identify strains with broad activity. So, we go for the development of BCAs at the next stage, where characterization of the selected strains are very, very important. That involves identifying traits that contribute to their effectiveness as biopesticides, and includes the production of antimicrobial compounds and traits related to colonization and nutrient uptake. Genome sequencing and comparative genomics offer insights into biocontrol mechanisms and potential new genes. There are pilot trials in different conditions which ensure the BCA's efficacy and consistency. Then the final development of bacterial control agents is the formulation and delivery for commercial use. This focuses on large scale production, formulation, preservation. Effective production methods should maximize cell yield without altering strain characteristics, because if the characteristics are altered, the outcome will be altogether different and maybe not beneficial. Formulation, whether in dry or liquid, is key for shelf life and efficacy. Additives and adjuvants improve survival and also the ease of application. Biosafety studies ensure safety for plants, non-target organisms and humans. Monitoring methods track the microorganisms' behavior and quality during production. Microorganisms with generally regarded as safe status facilitate the registration process due to their safe use history. So, this table lists some of the recently completed genome sequences of potential microbial biocontrol agents, which are published in and after 2015. There are many, like Trichoderma virans, and you can have these accession numbers with which you can mine them from the biological databases. Then you have in Trichoderma so many different species, Hargianum, then Gamsi, then you have Ricci, and then Hamatum and so on. Then in Bacillus you have Cereus, Atrophius and Valegensis with different strains, and then you also have Pseudomonas fluorescens and other species of Bacillus subtilis here with different strains. So, with this we come to the end of our lecture. Thank you for your patient hearing.